Hello, I'm Graham Chedd with the Alan Alda Center for Communicating Science here at Stony Brook University, and this is another episode of Science on Tap. Actually, it's one of a rare occasions when we're not in our bar because it's booked today, but we have a distinguished visitor to campus that we didn't want to miss the opportunity to talk with. And that's certainly the case today when our visitor is Dr. Donald Ingber, who's the founding director of the Wyss Institute for Biologically Inspired Engineering at Harvard University. Now, I could go on for about 10 minutes listing all of Dr. Ingber's accomplishments and papers and companies that he's founded, but I'm going to let you turn to Wikipedia for that, because I want to spend the time hearing from him rather than hearing about him. So I'm going to start by taking him back to his days at Yale, when he had an unusual launch into the field that he's in now, because it came about not through a science class, although science class contributed, but through an art class. You signed up for an art class. Tell us why and what happened.
So I grew up on Long Island, not far from here, East Meadow. I got into Yale when I was 17. I got in on math as it was my forte, although science and math were my interests. But when I got to Yale, I saw this incredibly rich environment for the humanities, for arts and so forth. And as a child, I always loved art, but I had no contact with it. And so I always tried to get into art classes just because I wanted to expand my horizons. But it was almost impossible at Yale. There was so few courses and so many art majors that they literally would have interviews through the night. I remember interviewing at 2:30 in the morning for a painting class and didn't get in. So I went on, and first year I didn't take much science. I did humanities, but then eventually I got into science and got excited about it. And I was in molecular biophysics and biochemistry. This was in the mid-'70s, early to mid-'70s. This is before the people had moved to higher organisms in terms of genetics and genomics. It was sort of basic organisms. And in those days, molecular biology was the biology of molecules, DNA, enzymes. And in my courses, everything was physical. It was the shape of the molecule. It was the double helix, that it actually was a helix, and the complementarity, that where the information lay. And I remember seeing these kids walking around campus, coming out of the sculpture hall, which was not far from where the science campus buildings were, carrying these sculptures that were kind of looked like viruses to me, and I would say, like, what are those? And they saw the sculptures, and I said, what course is it? And they said, it's a sculpture course called three-dimensional design. And to me, I was learning in molecular biophysics that everything was three-dimensional design. Now, the way I got into that course was serendipity, which I find is, in science, serendipity happens all the time. You have to learn how to see it, notice it, and act on it.
Do you mean things that come up unexpectedly,
right.
but you take
By
advantage
chance,
of?
Right. So it turned out that I had a girlfriend at the time who was a sculptress, and she was taking a human figure class with the same professor who taught three-dimensional design. I had no idea about this. So she said, would you like me to introduce you to him? So I said, sure. And he was this hulk of a man. His name is Erwin Hauer. He was like 6'2", big, had hands that had been working and sculpting for his whole life. I think he had a wooden leg from World War II. I think he was in a concentration camp or something along those lines, but he was terrifying. And I went to him, and he said, why do you want to be in my course? And I said, well, I'm a scientist, and your course is three-dimensional design, and everything is three-dimensional design at the molecular. I said, what do you mean? And I started to describe, DNA is a helix, and the information's in it. And he says, what do you mean? And I said, well, the muscle, it has actomyosin filaments. They're almost like screws that would move sort along each other and shorten. And he goes, in my course. And then I kept going, well, then there's this. And he said, in my course, just come.
Oh, so he invited you.
So then he invited me to the course. So I came the next week. And just a quick aside, by this time I was doing science, and I had done a summer on a fellowship in England where I had done cancer research, and I had come back, and I was learning how to culture cancer cells, first time in my life in the medical school, which is the complete opposite side of the campus at Yale. And when you culture cells, they're in a dish, they're kind of flattened. You add a chemical, it's an enzyme that clips the anchors of the cells to the dish. And the cells were flat, but when you clip the anchors, they just round up, ball up, float, and now you take them and you put them on another dish where they then reattach and spread. So this is literally 2 days before my art class, I saw that. In the art class one day, the teacher comes in, and basically he had told us to buy wood dowels that were 3 feet long and high tension fishing line, which we did. We came in with it. He brought us to a machine shop. He cut the wood dowels into 3 foot long pieces, and he made little X's at the end where you could tie the strings. And then he brought us all back to a big open room, and he said, I want you to build structures that hold their shape in 3 dimensions with the sticks and the strings, but the sticks can't touch. And he left. And you had all these kids, we were like 18, 19 years old, looking at each other, and people are like trying to hold sticks together, but they can't touch. And finally, one student who must have been an artist or sculptor who was familiar with Kenneth Nelson's sculptures figured out how to build one of these. And basically, the sticks are interconnected by a continuous series of strings so that when you put the last one in, it sort of pulls itself open. And then once that happened, it was like a wave of creativity. Every student started building different shapes and sizes and spirals. It was amazing. He came back.
Did you build one?
I did, and it's really funny because I just, a month ago I was in Berlin. There's a museum called the Martin Gropius Design Bau Museum, and that's in a glass enclosed exhibit, as is my cell model. But yes, I did. And
now these things sort of bounce, right?
Well,
They're springy?
the first one we built was with non-extensible high-tension fishing cables, so they were very strong.
Mm-hmm.
But the teacher afterwards gave us a little slide lecture on tensegrity, which is this build comes from tensional integrity, tensegrity. And he had a little structure that was round with elastic cords instead of non-extensible fishing. And as he spoke, it was round. He would just flatten it, and he'd let go, and it would bounce up off the table. This is totally true.
Reminded you of something.
And then, so I immediately, this was about 1975, and the first papers I was reading on cell shape, because we're using drugs that affected the shape of the cells in the cancer lab, we're talking about a cytoskeleton. It's a molecular framework inside a cell. At the molecular level, that's like our skeleton. And I remember going back to the cancer lab, and we added a drug, and the cell rounded up. And I said, oh, the tensegrity changed. and the postdoc said, what do you say? And I said, the tensegrity changed. And he said, what's that? I said, well, it turns out that's Buckminster Fuller first coined the word, who invented the geodesic dome. It's how the geodesic dome gets its stability. And then I was telling him about the sculptor, Kenneth Nelson, who was a student of Fuller, who first built one of these. And I learned it in a sculpture class. And he said, never say that again. and so basically, I shut up. And I went to the libraries at Yale. And I was an undergrad, and I had done all these different areas. So I would go to the art library, the science library, the math library. And I began to see that people in the 60s had talked about structuralism, and artists, writers, Buckminster Fuller, and they were talking that the answer is not in the components, but in the networks, the arrangements, the collectives, and that new functions emerge from collective behavior, and that this kind of embodied that. And so in any case, the net sum was that I saw these two things and immediately said, oh, that's how cells are built. Went back and said, shut up. And then I realized I was right, and then the rest of my life was kind of, my PhD was actually demonstrating that was true by building models of cells with nuclei, and those are in this museum now as well. They had conservators take my 40-year-old sculptures, which were in a closet, and rebuild them, which was really exciting for me.
So do cells literally have those sort of structures inside them?
So we do too. I mean, so our bodies, if you go to an anatomy lab and you see a skeleton, it looks like us because they wire the bones together and hang it from a stick. In reality, we're 206 compression-resistant bones, which I know because I learned it in Jeopardy when I was about 8, 206 bones, and that are interconnected by a continuous series of of tensile muscles, tendons, ligaments, fascia, that when we pull ourselves up against gravity by tension in our muscles. And like, I can be very stiff or flexible by the tone in my muscles, that engineers call pre-stress. Tensegrities are structures that gain their shape stability by pre-stress in the strings, basically. And now the cells, we had to show this over 30 years experimentally, which led me to develop entirely new methodologies. But yes, cells have hollow, stiff filaments called microtubules. They have contractile filaments that are made of actin and myosin, just like in muscle. They have the nucleus is at a smaller scale, a structure built the same way. It's connected by other filaments to the cell surface and the other cytoskeleton by intermediate filaments. And when you spread a cell, the nucleus spreads in a coordinated manner because it's kind of hardwired. And I built models in my thesis with a nucleus, a cell, and I put black elastic thread and a black background. You couldn't see it. And when the cell spread, the nucleus spread. And so, yeah, cells are built that way. And we showed in a science paper that we had to develop methods to measure the shape stability of a cell, the mechanics, how stiff it is. But it's dependent on pre-stress in the actomyosin at the cell level, just like my muscle at the organ level.
How did that insight into how cells are structured lead on to a much wider field that you're now in, which I love the title of biologically inspired engineering. It's sort of self-explanatory, but needs more explanation.
Yes. So, first of all, when I was in my undergrad courses in molecular biophysics, everything was explained physically, the physics of molecules, how the lock and key of an enzyme and a substrate, the helical DNA shape. You can make DNA out of other molecules, but if they take that shape and the complementarity, you could have information storage. It's not the chemistry, it's the spatial relationship. So everything was mechanics at the molecular level. I stay at Yale in graduate school and medical school. I did an MD, PhD, and I learned about the biology of cells the way it's taught. And it's all chemistry. There's no mechanics. And it was like hard, it's made of molecules. You group them together, now you have an organelle and you have a cell, and there's no mechanics anymore. So what in all my reading, and also I did developmental biology, how embryos, organs form in the embryo. 100 years ago, it was all explained mechanics, in mechanical terms and physical forces. But once chemistry came in, genetic, it all became reductionist, and it's like what it's made of. But they were able to explain things that we still can't mechanically. So there had to be an interplay between these two. So tensegrity allowed me to realize that it's a way that you can convert mechanical forces into changes in rearrangements of structures, whether it's if I push on you and your body reacts, or down to cellular scale, down to molecular scale. And once you put a force on a molecule, I knew from my molecular biophysics, you change biochemistry. You change gene expression. You can pull on a motor and change how it moves. You can pull on an enzyme and change its activity. You can pull on a molecule in your connective tissue and unfold it, and it polymerizes in the lines of force. So tensegrity provided a way to explain how you convert mechanical information into changes in cellular biochemistry and gene expression, which I called, and now we call mechanotransduction. And that whole field came out of mechanobiology. Okay, so that's one thing that is core to what I do, this idea that mechanical forces may be as important as chemicals and genes for regulation of biology. Now, about 8 years ago, I formed the Wyss Institute for Biologically Inspired Engineering. The concept with that is that we've uncovered enough about how nature builds, controls, and manufactures it from the nanoscale up, that we're now at a point where we could leverage biological principles to develop new engineering innovations. Bioengineering transformed medicine over the past 50 years. People hear about genes and genomics, but really what changed it are things like, hip implants, pacemakers, stents, artificial limbs, drug delivery. Those are all engineering improvements, billion-dollar industries each. And genomics is, diagnostics, maybe beginnings to see, but the last 50 years was engineers taking engineering principles, trying to solve medical problems, and it worked great. What we're saying, we could flip that on its head. Now we know enough about biology that we could leverage biological principles to develop new engineering innovations, and I call it biologically inspired engineering as opposed to classic biomedical engineering or bioengineering. And then tensegrity and mechanical forces is a design principle, and so we've done a lot leveraging that.
Let's take a little side trip. What things in nature have evolved structurally that impress you? I mean, I've always been impressed by a feather. It's an astonishing thing, with so light and so strong. What other examples are there in nature that inspire not necessarily particular developments, but just awe?
I mean, I'm in awe of everything in biology. I see the hierarchical self-assembly. So, I see how life evolved using similar rules, spatial constraints, this sort of tensegrity, collective behavior. So there's, first thing is this idea of, I had a Scientific American where I called the architecture of life. Okay, so tensegrity explains geodesic structures. If you start in the inorganic world of crystals, they're all geodesic. Geodesic means shortest path. And so when you have like carbon atoms or other atoms, sodium chloride, they're pulling and pulling until the nuclei can't be compressed, and then they're stable and rigid. That's a tensegrity structure, but it's incredibly rigid. The first time you see the first step towards living systems is when you get to clay, which is where people think some people think life emerged in clay.
Right.
Clay has these very rigid, let's say, like triangulated tetrahedra that are stiff, but then instead of being packed triangulated, there's like a pentagon or hexagon, and they could wiggle around the pentagon. That can now catalyze chemical reactions. People showed you could add amino acids that may come on meteors, and you get polymers. You get the first peptides. You get the first RNAs. So then these proteins and polymers start folding up on themselves by attractive bonding tensile forces, but their own stiffness in locations prevents them from being a crumped ball. That is the first tensegrities at the molecular level. So to me, I see this going like through the first simple molecules to multimolecular structures, the first simple organisms, to multiple cells, to sponges. So to me, it's amazing that this simple principle guided, even, people think DNA drives evolution, but we evolved before DNA came into existence. So it was these spatial constraints that drove evolution, and then DNA turbocharged it, because now you could explore. So, I mean, I love the beauty of patterns. So I also, we have one person, I do organs on chips now with microfluidics. I have a technician who put sea sponges in there, and they produced to try to model production of artificial glasses, and these beautiful structures that are all perfectly regular. I sort of see beauty in all natural systems. I can't give you one, but if you ever watch an embryo develop, it's pretty awe-inspiring.
Yeah. So let's talk about organs on chips.
Yeah.
Where did that idea come from, and what do they do?
So this is, if you want the real story, it's a little bit of a long story, which is I believe that mechanical forces were important. I wanted to show that if that's true, if you were to physically stretch or distort cells, that should change their behaviors. I did it various ways, but people kept criticizing that I was getting cells to, took a dish that cells couldn't adhere to, and I put many molecules that are called matrix molecules. These are anchoring molecules in our body. They form, I call it the egg carton that eggs sit in. Our cells sit in an egg carton that's called extracellular matrix. And when I did that, to the degree the cells spread, if I put more molecules, they spread more or less. They were round. When they spread, they would proliferate. When they were round, they would go into a suicide program. When they were in the middle, they would differentiate. Capillary cells would form tubes. Liver cells would produce liver protein. I thought that was a pretty great example that shape and distortion regulated biology. Same growth factors the whole time. They said, well, it's not the shape, it's you have more matrix contacts. So then I was thinking, how do you get around that? And this is a lot of science. It's like listening to the smart critics and say, how do I convince them? And so I wanted to make little islands that had a high density of these matrix molecules. Same density, a big island surrounded by a non-adhesive Teflon-like region, where if this, let's say it's round and the cell spreads, it spreads by binding and pulling and pulling and flattening, and you get a pancake-shaped cell. And then I would make a smaller island, same density of matrix that they think is so important, and I have a cupcake-shaped cell. Then I have a little dot, I have a golf ball-shaped cell, and the only difference was shape. And that's a science paper called Geometric Control of Cell Life and Death, which has 4,000 citations. And I did that by collaborating This is another serendipitous thing. I was working with etching with lasers and glass to make islands, and the grad student was in a bar with another grad student of George Whiteside's, and he learned that George Whitesides, who's a famous chemist at Harvard, had just developed an inexpensive way to make microchips for the computer industry. Computer microchips use stereolithography with a silicon chip, and then they have a mask that with ultraviolet light, and they make little lines and patterns. Everyone's incredibly expensive. What he did is he would etch a chip that's sort of the reverse pattern, and then he would take a liquid polymer that we polymerize, and it's rubbery when it polymerizes, and it's optically clear, and he'd peel it off, and he'd have a rubber stamp. And then he would stamp chemical inks onto other surfaces that were in that same pattern down to nanometer resolution, and you can make 1,000 chips. So they realized, the students said, well, maybe this would be useful for what we're trying to do, is we could stamp patterns of this matrix. We did that, it worked, and that's made me realize that computer microchip manufacturing has control over the same features scale, the nanometer to micrometer, that cells live at. And so we were creating these artificial culture environments. Go a couple years later, George started to use these to make what are called microfluidic systems. These are the same technique, but instead of making a pattern of dots or circles or lines, you make an elevated ridge, and then you polymerize around it, you take that off, you put it on another surface, and now you have a tunnel, a hollow tunnel. And you could have little tributaries coming into it and joining into a bigger tunnel, and then outlets. And that you could flow liquids through. And that was used by George and many others to miniaturize instrumentation. And that has taken refrigerator-sized machines and made them into iPhone-type machines. And I'll give you the full story. So one of the cool things about it is that if you were to take two different hoses filled with, two different colored fluids and flow them into one little river, they would mix. It'd be turbulent, like cigarette smoke mixing in the air. But if you go down below about 1 millimeter, you only have laminar flow. There's no turbulence, because turbulence, if you go back to high school physics, is a function of the radius, how wide it is. And so what happens is you put a red and a blue, and they just flow right by each other, red and blue, and then they flow out. And so I went to medical school. I'm always thinking about application. I looked at that and I thought, people were using it for diagnostics, but I thought, could that be used therapeutics? And I thought, well, what if you had blood and sterile saline? They wouldn't mix. And then maybe you could pull things across it that are bigger than a dialysis pore, which is minuscule. You could have a small molecule like a glucose, but you can't get cells or protein. Maybe you could pull it across it some way. And then I thought, well, how can we do that? And I thought magnetically, because I didn't talk about this, but we used magnetic beads coated with these anchoring molecules, matrix, to pull on cell surface receptors to show that cells can sense force and to measure how stiff they are. So I had all these different methods to keep folding back in a creative way. And that led to what we called a spleen on a chip. Now, this was not what we call organs on chips now. It was a conceptual spleen in that it would cleanse blood of pathogens with people that had sepsis. Side story there, with the Wyss Institute, we've optimized that, developed ways to we engineer natural proteins to capture pathogens without knowing what they are, which is critical, because people with sepsis, 70% are blood culture negative. They never know what the bug is, and that's why the mortality rate's so high. So we engineered a protein that is part of your primitive immune system that were before antibodies that capture bugs based on carbohydrates on their surface, and it turns out this one protein, it's called mannose binding lectin, MBL, binds over 90 different pathogens of every class, toxins, viruses, Ebola, antibiotic-resistant organisms, gram-negative bacteria, gram-positive, fungi, etc. We engineered it, and we put it on these beads, and we could pull bugs out, and we could save animal lives. And then we are now commercializing that in a company. That's one of the startups, and we hope to be in humans within the year. But now when you start a company, it's a different world. You work with the FDA, you work with cost, you work with regulation. And we realized that the beads are totally new, there could be complications. So we just took the protein that we engineered through that serendipitous path and put it on an existing dialysis cartridge that's FDA approved, and it works pretty well. And so that's what's going to humans.
Right.
So going back, so we had spleen on a chip, not organ on a chip. I go to a meeting, and a student of mine and George Whiteside's, a postdoc of George's who worked with me on microfluidics, had gone on to his own career. His name is Shu Takayama at University of Michigan. And he gave a talk. We talked about a lung on a chip.
Lung on a chip.
Lung on a chip. But it had no cells, but it had these channels. They were the size of the small airways in a lung. And he was putting little droplets of fluid through to mimic mucus plugs that people have when they have pneumonia or sick. And what blew me away is that he recorded a noise that came out of this little chip. And that noise is exactly the noise, and he did an analysis, exactly the noise that I was taught as a medical student to listen for through a stethoscope, which is called a crackle. It sounds like if you rub your fingers next to your ears. And if you hear that, that means fluid on the lungs. And you'd always ask the professor, like, what is that from? And they say, I don't know, fluid in the lungs, I don't know. And this showed it was exactly little plugs going through and then popping. So the grad student who did that, Dan Ha, applied for a postdoc with me, came to my lab, and I said, that is fantastic, but why don't we build a real lung on a chip? And my idea was to take those two solutions that don't mix and to form a membrane at the interface, which would be this extracellular matrix egg carton that cells sit on, and put the lungs lining cells of the air sac on one side and the capillary blood vessel cells on the other, and we'd recreate, and then we'd put air through one side and we'd recreate what's called the alveolar capillary interface.
Is it with real cells?
With real cells. That was the idea.
Yeah.
And long story short is that we couldn't polymerize very well, but he made an artificial membrane with pores coated with matrix, and it worked. And that was the real first organ on a chip, lung on a chip. Long story, but it's the true story. That's how.
Now, do you now have a lung on a chip, a spleen on a chip, a kidney on a chip?
We have over 10 different organ chips.
Okay, and even if you're
and we've linked them together.
A body on a chip.
Yeah, human body on chips.
Now, what's the use of these things? I mean, they're fascinating, but are they valuable?
So, when we started the Wyss Institute, we had the largest gift in Harvard's history at the time, $125 million, and we've doubled that since. And we were supposed to take on high-value, high-risk problems that would bring about transformative change. And the biggest problem that I could see is that the drug development model is broken. You basically go to the CEOs of these big pharmaceutical biotech companies and you hear their vision, and it's to fail quickly and cheaply. Because they fail so much, they would rather fail earlier than going to all the way to clinical trials where they fail all the time. And a big part of the problem is that most of the studies are with cells in a dish, on a plastic dish that look nothing like our body, and then the other preclinical studies are animal studies, which they have to do to please the FDA and to do safety and efficacy. And a mouse is not a man. Everyone knows that. The drug companies, biotech, they know it, but they say we have to do it for the FDA. The FDA says we don't think it's good either, but we have nothing better. And
so many drugs work in mice, but when they get to the next stage, they don't.
In some areas it's 70% failure. I give a talk, and I show two slides. One is Moore's law, which is this exponential growth of packing in power density into microchips over 50 years. It's linear on a log scale. And the other comes from a paper that's not my work. They call Erwin's law, which is Moore's law backwards, which goes down since 1950 exponentially the number of new drugs approved, even though exponential increase in funding. So there's a big problem. So these are human cells we use in these chips. We are building sort of major functional units that are really organ, because organs are two or more tissues that come together when new functions emerge. So we have the tissue of the airway cell, tissue of the blood vessel, and we form an interface. We can flow blood. We flow medium with immune cells, and now we flow whole blood through the vascular channel. We could put air. If it's a lung, if we have a gut, we could have fluid. We could have peristaltic-like motions. I didn't say for the lung we had breathing motions because I thought mechanics is important. So this is made in that same flexible, clear rubber that we made rubber stamps with. And you have the channel. You could think of like a Holland Tunnel in New York, or a tunnel. There are actually 3 tunnels. The middle tunnel has a horizontal membrane that has pores that we coat with these anchoring proteins, the matrix. On the top, we put the lung airway cells or alveolar cells, and we put air. On the bottom of the same membrane, we put the capillary cells, and now we actually cover all 4 walls with capillary cells. And then you could flow medium through, and that feeds the organ like it would be fed in the body. But the trick is we have side channels that have cyclic suction. And because this is all flexible and very, very thin-walled,
it bows out when you apply suction and it comes back. So that you could actually have it cyclically extend and contract just like when you breathe in and out in your alveoli. And with our gut, we do the same thing to mimic peristalsis. And amazingly, with the gut on a chip, if you take cells that people have used for 50 years that are from human intestine, they always grow as flat cells on a dish, poorly differentiated. Whereas in our body, they're columnar, they're tall, and they form villi in our intestine. If we give them flow and peristalsis, they become columnar and they form villi in 5 days.
Okay, so you can make these little
Yep.
model organs. How is it useful for drug testing?
Oh, so basically, we showed, for example, one is toxicity. So we used the lung on a chip, and well, the big problem is disease models, because people know that animal disease models don't look like human disease. So we tried to hit two birds with one stone. We did a disease model and a drug toxicity model by putting a drug called interleukin-2, IL-2, which is FDA approved for cancer, but its dose-limiting toxicity is fluid on the lungs, pulmonary edema, and pulmonary edema is a disease. So what we did is we injected the drug intravenously, like in a patient, but we put it through our vascular channel, and we saw at the same dose, same time course, fluid filling the lungs in 2 to 4 days, just like in humans. Then, so now we had shown a drug toxicity we could mimic, and we basically had a model of a disease. Now, I talked about my mechanical work. And so in the years of pursuing how cells sense mechanical forces, we found out that one of the first thing is we found that cells don't feel force equally all over the surface. It is like a tensegrity structure, or let's say a pup tent. A tent has a membrane, and if you want to hold it in a shape, you put sticks up, which are the struts in a tensegrity, and you tie it, put pegs in the ground, and you winch it in under tension to hold it. That's a tensegrity. So the tent doesn't feel forces at all points. It feels it through the tent pegs. Well, that led me to predict that cells would feel forces through receptors that link the extracellular anchoring sites to the cytoskeleton, and those are called integrins. I predicted this before people discovered integrins, but then once they found them and we had probes for it, we had another science paper where we used magnetic beads coated with ligands. If we pull on integrins, you feel the whole strength of the cell, you can move things inside of it, and the cell behave mechanically like one of my tensegrities. We have a picture of a tensegrity and the cell, and we measure mechanics with force, and they're identical in the science paper. We also could show that when we pull, we change biochemistry and gene expression. So that turned out to be true. But the fastest conversion event was when you pull on these receptors called integrins. Right near the integrin, there's an ion channel called TRPV4 that lets calcium in the cell. And that triggers downstream signaling. And I had heard, and so remember, I didn't say this, when we found this pulmonary edema with interleukin-2, we got it with breathing movements when it was physiological. But when we stopped breathing movements, it was down only a small effect. So breathing movements were absolutely critical for this effect. So that's validated that breathing is important to mechanics to mimic organ level physiology. Mm-hmm. So I heard GlaxoSmithKline was working on an inhibitor of that ion channel. We got it from them. They didn't fund us. We just collaborated. We tested it. It prevented pulmonary edema in the chip. They then went and tested it in animal models of pulmonary edema, and it got the same thing. And then we had back-to-back papers in a journal, a high-impact journal. And that drug's in phase 1 clinical trials. Before that, the program was kind of a question of whether it was even going to go on. So that's examples of drug toxicity, drug development. Now it's gone much further, and we work with other companies. There's a company called Emulate we founded, and they're working with multiple drug companies. They announced a collaboration with Janssen Pharmaceuticals. They have a drug that had no toxicity in animals, and it failed in human clinical trials with pulmonary thrombosis, clots in the lungs, and we can mimic that on the chip. So those are examples of how of what could be done. The other more exciting idea, I think, is personalized medicine. So, and sort of a nuance on that. Obviously, I could make a lung chip with your cells and test drugs for you, and that's a possibility, especially now that we have induced pluripotent stem cells where you could take a skin cell and make it into any cell in your body. But I think the more powerful idea is that right now drug companies spend, there's a paper 3 years ago from Tufts, $2.6 billion to take a drug from discovery at the bench to clinical approval. they will spend a huge amount of money in the early phase. They go to human trials. They'll do a big trial, and they almost always fail. And what they do is they'll collect the data, take a year with statisticians, and try to see is there a genetic subgroup that maybe responded better, and then maybe do a small trial and get approved. With organs on chips, you can imagine making chips from genetic subgroups from the beginning. So let's say you have Hispanic, female, asthmatics who are ultra-sensitive to cigarette smoke. And we now could apply cigarette smoke directly to airway chips with a little cigarette smoking robot. And so let's say we get 100 of those women. We get their cells either through a bronchoscopy or IPS, induced pluripotent stem cells. We make the chips. We test drugs for that group. We find something. We use those patients as our clinical trial. We have a small clinical trial. We get approved for that. That could revolutionize how drug development is done. That, to me, that is the game changer.
Mm-hmm, mm-hmm. One of the neat things is that your organ on a chip is now in the Design Museum in London, right? And it was the first time ever a medical device won a design award.
Well, it won it won 2 awards, the best product and the design of the year last year at the London Design Museum. It also was acquired for the permanent collection of the Museum of Modern Art in New York, which to me is even more amazing. I'm now a permanent member of the Museum of MoMA. Yeah, and it's interesting. It's in multiple museums right now. It's also in the Martin Gropius Museum. It's in Copenhagen on display. It's all over the place. It
must be very satisfying seeing art sort of started you.
It's unbelievable.
And now it's gone back into the art world.
I gave a lecture, a class day lecture at Dartmouth Medical School a few years ago, and about life, talked about life path, and I said, my life is a biased random walk. A random walk is like molecules move randomly. Cells move randomly. But if you give a cell a stimulus that it's attracted to, it moves randomly, but it just always tends to go a little bit more towards what it wants to be, and it ends up there. And that's life. You kind of have to go all over the place following your passion. But if you really follow your passion, you tend to edge that way, and somehow it's amazing to me, incredibly gratifying. So the organs on chips are a leap forward in design, in the sense that, and Buckminster Fuller was actually a big developer of design science, the idea that design could have positive impact on the world, which is really being embraced by the design community right now. It's not just aesthetic, it's actually design that empowers, that's impactful. We didn't build a lung. We didn't do tissue engineering to build a lung. We tried to distill it to its minimal design principles, like what makes an organ, 2 or more tissues that come together and new functions emerge. I believed that the physicality was important, so we had to have the breathing motions. We then went to man-made approaches, computer microchip manufacturing, as a way that could be potentially inexpensive, scalable, cheap, and robust. And we kind of combined all this together, and by chance we used simple patterns because it's easier to do symmetrical things. It's optically clear, it's almost crystalline, and it's kind of beautiful. So it is, but it is design. I mean, we have a patent, and the pictures in it are design diagrams, effectively. So yeah, no, that I'm very proud of.
Now, you've given a lot of public talks as well, right? Of like 500, another one coming up.
Yes.
Why? Do you think communicating your science is important?
Oh, science, I learned early on, you have to be, it's really a Sermon on the Mount. People don't really read. And if they read, they only read, it's like the media, they read what they already believe in. They don't read new ideas. But if they hear you speak and they get excited, they'll go back and say, I heard this guy, and then they'll look it up afterwards. So you also have to meet people. When you go to meetings in science, you meet people, you're really impressed with them, you trust their work. You meet people, you're not impressed with them, you'll never trust their work. It's a people business. One of the travesties about science, I say this all the time, is that it's taught as memorization, where by definition, if you can memorize it, it's not science to a real scientist. Science is exploring the unknown and defining the unknown. If you can memorize it, it's not science. Yet that's the way we're taught in school. And I think we scientists have a responsibility science is the only hope, not only for the world, but for the economy. I mean, technology is what made America lead the world. Optical fibers, computers, medical devices, that came from science. It's not like, oh, we'll fund, startups. Startups come from science. And so I think we have a responsibility, if you want to have a positive impact, to make people realize that science is a creative discipline for those who think it's not, that it's a impactful discipline who think it's purely, in a white tower, and that there's the beauty of understanding how things work. And there is a Judah Folkman in a Nova interview. I worked with Judah Folkman for years, who was really a brilliant, brilliant man. And he said, there's no feeling like the high of realizing you now understand something that no one in the world has ever understood before. And I've had that with tensegrity. I've had that with many other things. And it is true. It's like a long distance runner who can't stop running. Once you feel that, you can't go back. I think it's important to communicate that kind of appreciation for the beauty, the aesthetic of science, the visual beauty of the feather or an embryo or it's wonder. You're basically able to spend your life like a small child. It's one of the few businesses where you can be an artist, a small child, but actually make a living. But it's not taught that way at all. So I feel like we have a responsibility to do that.
Well, not only were you biologically inspired, but you are inspiring. And thank you so much for coming to tell us about your work.
Oh, thank you so much.
Congratulations.
Thanks. Thank you all.